Welcome to the Explainer. So, let's start with a really big question. How does life build itself? I mean, really, how does a single cell become, well, you? It's this incredible puzzle, right? And the answer is hiding in plain sight, inside a kind of secret code. And today, we're going to crack it.
I mean, just stop and think about that for a second. You started out as just one single cell. So how on earth did that one tiny cell know what to do? How did it know how to become your brain, your bones, and your heart? Well, it turns out there's a master plan. It's this beautiful set of instructions that's followed with just mind-blowing precision inside every single one of your trillions of cells. And this simple little diagram, this is our roadmap. This is the heart of the whole thing. It's called the central dogma of molecular biology, which is a fancy way of saying it's the basic story of how life works. You've got information, right, that's stored in DNA. That gets copied into a temporary message, which is RNA. And that message is then used to build, well, everything, all the machinery of the cell, which we call proteins. So let's unpack this step by step.
All right, first up, the blueprint of life. It all begins with DNA. You can think of DNA as the cell's master blueprint, okay? It has every single design you need to build and run an entire organism. And just like a priceless architectural blueprint, you absolutely have to copy it perfectly every single time a cell divides.
So how does the cell pull off this perfect copy? Well, it uses this really elegant and frankly clever method called semi-conservative replication. Here's how it works. The original DNA ladder unwinds, right? And each of the two old sides becomes a template to build a brand new partner. So what you end up with are two new DNA molecules, but each one is a hybrid. It's half old, half new. And this is just a genius way to cut down on mistakes and make sure every new cell gets a perfect, identical copy of that original blueprint.
Now, this whole process, it's not magic, right? It's more like a super efficient construction site run by a whole team of these tiny molecular machines we call enzymes. So let's meet the crew. You've got helicase, we'll call that the unzipper. Its job is to pry open the DNA. Then you have primase, the starter, who comes in and lays down a little foundation to get things going. Then the star of the show, DNA polymerase, the builder. This one adds all the new DNA bricks one by one. And finally, you've got ligase, the gluer, who comes in at the end and just seals everything up to make sure the new strand is perfect.
Okay, but there's a little catch with this whole copying thing. It's a tiny problem, but it's a big deal. Every time DNA gets copied, a little tiny piece at the very end of our chromosomes can get, well, lost. To stop this from happening, our chromosomes have these protective caps called telomeres. You can kind of think of them like the plastic tips on a shoelace, or even like little molecular clocks. And there's a special enzyme called telomerase that works to keep these clocks in good shape, making sure our super important genetic info doesn't get worn away every time a cell divides.
Okay, so we've got the master blueprint, the DNA. But here's the thing. These plans are way too valuable to just take out to the construction site where they could get damaged. So they're kept locked away for safekeeping in the cell's nucleus. Think of it like a master file in a central library. So when the cell needs to actually build something, what does it do? It makes a temporary, disposable copy of just one specific part of the plan. And that working copy, that's called RNA.
So what's the difference between this RNA message and the original DNA blueprint? Well, a couple of things. DNA is that famous double helix, right? Super stable. RNA is usually just a single strand, much more flexible. It also swaps out one of its chemical letters. It uses something called uracil instead of thymine. The best way to think about it is this. If DNA is the permanent, hard copy blueprint, RNA is like a quick, single-use photocopy or a memo. It's mobile, it's disposable, and it's ready to be sent out to the factory floor.
But before this RNA message can actually leave the library, it needs a little bit of processing. It's kind of like prepping a letter before you mail it. First, it gets a protective cap on the front end. Then there's this really important editing step called splicing, where all the junk parts, the non-coding bits, get cut out. And finally, a long tail is added to the back to make it more stable. Only after all three of those things happen is the message actually ready to go.
Okay, let's get to the final step, building with the instructions. So our finished, edited mRNA message leaves the nucleus, and it travels out to a structure called the ribosome. This is the cell's protein factory. This is where the magic happens. It's where the code gets translated from the language of genes into the language of actual physical stuff, proteins.
And here's something that is just so cool and really shows you how important RNA is. The ribosome, this amazing machine that's reading the RNA message to build proteins, isn't actually made of protein. Get this, it's a ribozyme, which means the machine itself is made of RNA.
So RNA isn't just the message, it's also part of the machine that reads the message. How incredible is that? So this whole translation process works just like a high-tech, automated assembly line. The ribosome chugs along the mRNA message, and it reads it like a ticker tape, but in 3-letter chunks. We call those codons. And for every single codon, there's a specific delivery molecule, a transfer RNA or tRNA, that acts like a perfect little adapter. It reads the codon, grabs the one right amino acid, and brings it over. Then the ribosome links them all together, one after another, building this long chain that eventually folds up into a complex 3D protein.
Now, obviously, a system this complex and this important can't just be left to run on its own. That would be total chaos, right? It'd be super wasteful and even dangerous. And that's exactly why cells have these incredibly sophisticated layers of management and quality control. They need to regulate which blueprints get used, when they get used, and, crucially, to fix any mistakes that might pop up in the master code. So let's look at a classic example of how this control works. In bacteria, there's a simple little genetic switch called the lac operon. It's super elegant. See, bacteria only want to make the stuff to digest milk sugar, lactose, if there's actually lactose around. It just makes sense. So when there's no lactose, a protein literally sits on the DNA and blocks the gene. Gene is off. But the moment lactose shows up, it grabs onto that blocker, pulls it off, and boom, the gene is switched on. It's just a really smart, efficient way for cells to decide which instructions to use and when, so they don't waste energy.
And what about keeping that master blueprint safe and sound? Well, cells have an entire quality control department just for that. These DNA repair systems are on patrol 24/7. You've got different crews for different jobs. Some are like a spell checker, just fixing typos that happen during copying. Others are like tiny surgeons that can precisely snip out a single damaged letter. And then you have the heavy-duty crews that can come in and cut out bigger chunks of damage, say from something like UV sunlight.
So when you zoom out and look at the big picture, you see it. This is the fundamental process of life. Information flows from that permanent DNA blueprint to a temporary RNA message, and finally into a real working protein. And none of this is random. It's this beautiful, highly coordinated dance of molecular machines, all kept in check by these amazing layers of control and repair. It's just an incredibly elegant system, and it's happening inside you, trillions of times every single second. This whole system is amazingly robust, but it's not perfect. It's not foolproof. The repair crews can get overwhelmed, those genetic switches can get stuck in the on or off position, so that leaves us with a really important question. What happens when this elegant, precise code of life fails? What happens when it all goes wrong? Well, that's a thought for next time. Thanks for joining this explainer.